Right now, before this explanation begins, I want you to notice something you are probably not aware of. You are breathing. You know that, but I mean the specific rhythm of it, the slight pause between inhale and exhale. The fact that your diaphragm just contracted without you deciding it should. Your body is performing roughly 20,000 breaths today, and you will consciously notice fewer than 50 of them. What you do not notice is what is happening inside your skull during one of those breaths. Because there is a second rhythm run underneath the one, you can feel a slow, pulsing wave of fluid move through the spaces between your brain cells. It has been happening since the moment you fell asleep last night. It is happening right now while you are awake, though at a fraction of the intensity. And the reason it matters is that this fluid movement may be the single most important biological process you have or heard of. The discovery of this system is so recent that most medical textbooks printed before 2010, Single Fit 5, do not contain a single mention of it. In 2022, a team at the University of Rochester Medicus, led by Dr. Maiken Nedergaard, injected a tracer molecule in a small trace of a mouse and watched under a 2-photon microscope as it flowed the brain along pathway that no one had ever mapped. The tracer moved not through blood vessels, not through the tissue itself, but along the outside of arteries, a dedicated highway of fluid that had been sitting in plain sight for over a century. Nedergaard called it the glymphatic system. The name is a portmanteau: glia, the support cells of the brain, and lymphatic, the waste clearance network that runs through the rest of your body. The implication was immediate and startling. Your brain has its own plumbing, and it only works properly while you sleep. To understand why this plumbing is necessary, you have to understand what your brain is doing right now. While you read these words, every neuron firing, every signal passing across a synapse, every memory being encoded or retrieved, each of these processes generates metabolic waste. The brain consumes about 20% of your body's energy, despite being only 2% of your mass, and that energy use leaves behind a biochemical residue. The most studied of these waste products is something called amyloid beta, a peptide fragment that accumulates between neurons. In healthy brains, amyloid beta is produced and cleared in a daily cycle. In Alzheimer's disease, it accumulates into sticky plaques. For decades, research assumed that the brain's brain simply recycled its waste through the same systems as the rest of the body, or that the blood-brain barrier handled clearance passively. Neither assumption was correct. The glymphatic system is the brain's dedicated janitorial service, and it runs on a cycle tied directly to your sleep-wake rhythm. The mechanism works like this. Your brain is bathed in cerebrospinal fluid, a clear, colorless liquid that fills the ventricles and the subarachnoid space. CSF has been known to anatomists for centuries. Its functions were thought to be limited to cushioning the brain against impact and maintaining pressure. What Nedergaard's team discovered is that CSF does not just sit there, it flows. And the driving force behind that flow is something unexpected. Your own height, combined with the slow waves of the breathing cycle, pumps CSF from this brain cycle, pumps CSF from the spaces around the brain into the brain tissue itself. But it cannot simply diffuse through the dense mesh of neurons and glial cells. It needs a conduit. The conduit is a network of channels formed by cells called astrocytes. Astrocytes are star-shaped glial cells that were long considered mere structural support, scaffolding that holds neurons in place. What Nedergaard found is that astrocytes express a water channel protein called aquaporin 4, concentrate at the ends of their long processes where they wrap around blood vessels. Aquaporin 4 is a macroscopic gate that allows water molecules to pass through the cell membrane faster than they could by diffusion alone. During sleep, these channels open. CSF is driven in the brain along the outside of arteries, pushed through the astrocyte network by aquaporin 4, and then flushed out along veins, carrying metabolic waste with it. The entire brain is perfused with fluid, like a sponge being squeezed and released. The efficiency of this system is astonishing. In 2013, Nedergaard is in the journal Science, showing that the glymphatic system at Alzheimer at roughly twice the ring duration from the mouse brain, when roughly twice the ring sleep when compared to wakefulness. The volume of interstitial space the gaps between brain cells through which fluid flows increases by about 60% during sleep. Your brain physically expands its drainage network while you are unconscious. It is as if a city's entire sewage system only opened its pipes between midnight and 6 in the morning, and every other system in the city had to wait. But the discovery raised an immediate question that the Rochester team could not answer in their first papers. What actually triggers the switch? What tells the aquaporin 4 channels to open and the interstitial space to expand? The answer turned out to involve a second system entirely, one that had been studied for decades in a completely different context. It involves a molecule called norepinephrine, a neurotransmitter that your brainstem releases when you are awake, alert, or stressed. Norepinephrine is the chemical cousin adrenaline. It keeps your neurons firing, your heart rate elevated, your attention narrowed. It is the molecule of vigilance. And it turns out that norepinephrine is also the molecule your drains close. During wakefulness, the locus coeruleus, a tiny nucleus in the pons, no larger than a grain of rice, fires steadily, releasing norepinephrine throughout the cortex. This constant low-level signal keeps your neurons in a state of readiness, but it also constricts the spaces around your blood vessels. The astrocytes respond by tightening their grip on the perivascular channels. CSF cannot flow freely. Waste accumulates. The system is effectively locked during the day because the brain prioritizes awareness over cleaning. It is a trade-off that makes evolutionary sense. A predator does not pause to perform maintenance while scanning the savanna for threats. When you fall asleep, the locus coeruleus stops firing. Norepinephrine levels drop. The perivascular channels relax. The astrocytes loosen their grip. The interstitial space expands by 60%, and CSF begins to flow. The brain flushes itself. The entire cycle is governed by a single small cluster of neurons deciding whether to release a single molecule. And here is the detail that makes this system both elegant and fragile. The locus coeruleus does not simply turn off during all sleep. It turns off during deep sleep, the stage called non-REM, slow-wave sleep. During REM sleep, when you dream, the locus coeruleus fires again, norepinephrine returns, and the glymphatic flow slows or stops. Your brain cleans itself only during specific windows of the night, and those windows become shorter and shallower as you age. This brings us to the relationship between sleep depth and waste clearance, and why the modern sleep pattern may be systematically undermining the brain once ability to clean itself. A healthy young adult spends roughly 20 to 25% of the night in deep slow-wave sleep. That is the stage where the electroencephalogram shows large, slow delta waves, the synchronized firing of cortical neurons. It is also the stage where the locus coeruleus is quietest, where the glymphatic flow is strongest, where the brain is being washed. As you age, the proportion of deep sleep declines. By age 60, many people spend less than 5% of the night in slow-wave sleep. The glymphatic system runs for a fraction of the time it did in youth. Waste accumulates faster than it can be cleared. The implications for Alzheimer's disease are difficult to overstate. Amyloid beta clearance is impaired in aging, and the rate of clearance correlates, and the rate slowed directly with the amount of slow-wave sleep. Research at the University of California, Berkeley, led by Dr. Matthew Walker, have shown that a single night of sleep deprivation increases amyloid beta levels in the human brain measured by PET scan. One night. The effect is reversible with recovery sleep in young adults, but the trajectory over decades is unmistakable. Chronic short sleep, fragmented sleep, and sleep that lacks depth produce a net accumulation of amyloid beta year after year. The plaques that define Alzheimer's pathology may be, at least in part, a consequence of a drainage system that never had enough time to run. Now I want you to think about what happened during the hours you are actually asleep, but not in deep sleep. The average adult wakes briefly 15 to 20 times per night, microarousals that last a few seconds and are almost never remembered. Each microarousal triggers a brief spike in norepinephrine from the locus coeruleus. Each spike briefly constricts the glymphatic channels. The system does not need to be fully shut down to be impaired. It needs only to be interrupted frequently enough that the sustained flow of CSF never establishes itself. Fragmented sleep is functionally similar to short sleep in terms of waste clearance. The brain never gets the unrepeated block of slow wave time it needs to complete a full flush cycle. There is a study from 2009 that I find particularly striking. Researchers at Boston University, led by Dr. Laura Lewis, use electroencephalography and functional encephalography and functionality fragmented imaging to the relation of slow waves, blood flow, and CSF movement in sleeping humans. They found that slow waves, the electrosignature of deep sleep, are followed within seconds by a drop in blood volume in the brain, and then by a surge of CF flowing in the ventricles. The sequence is precise and rhythmic: blood outflow, fluid inflow. The brain is literally pumping blood out to make room for the washing fluid. The team could watch the CSF pulse in the brain in real time, synchronized to the slow oscillations of neural activity. What this means is that the glymphatic system is not a passive drain. It is an active, mechanically driven pump. The power comes from the brain's own electrical rhythms, coordinating with the cardiovascular system. Your heart pushes blood. Your heart pushes blood in the brain with each beat. The blood fills the arteries. And because the skull is a rigid container, something else must give way. That something is CSF. When blood volume rises, CSF is displaced out of the cranial vault. When blood volume falls, as it does during the slow wave oscillation, CSF flushes back in. The entire system is a hydraulic pump driven by the pulse. And the slow waves of deep sleep act as a gate that opens the path for CSF to penetrate deep into the tissue rather than just flowing around the surface. The scale of this movement is not trivial. Lewis' team measured that each slow wave-driven CSF surge displaces several milliliters of fluid, enough to be visible on the MRI scans as a dark wave washing over the brain. Over the course of a full night of uninterrupted deep sleep, the equivalent of the entire volume of CSF is exchanged multiple times. Your brain is washed from the inside out, repeatedly, every night when the conditions are right. When they are not right, when sleep is shallow, fragmented, or short, the wash cycle is incomplete. And this is where the story becomes personally relevant to every adult reading this. The modern environment is systematically hostile to slow wave sleep. Alcohol suppresses slow wave sleep, even in mod amounts consumed hours before bed. Caffeine blocks the adenosine receptors that help initiate deep sleep. Blue light from screens delays the melatonin onset that allows sleep to begin. Room temperature of about 65 degrees Fahrenheit reduces the likelihood of entering and maintaining slow wave sleep. Sleep apnea, which affects an estimated 30 to 40% of older adults, fragments sleep with hundreds of micro-arousals per night, each one spiking norepinephrine and closing the glymphatic channels. The average person in an industrialized country is subjecting their glymphatic system to conditions that would be considered unacceptable for any other organ's waste system behind. Waste clearance system. Now, I want to introduce the human story that made this discovery possible, because it is a story about persistence and about a scientist who was told her idea was wrong for years before it was proven correct. Maiken Nedergaard is Danish. She trained in neurobiology at the University of Copenhagen and later at the National Institutes of Health in the United States. For most of her early career, she studied astrocytes, the star-shaped glial cells that outnumber neurons in the human brain. At the time, astrocytes were considered support cells at best and biological packing material at worst. The prevailing view in neuroscience was that neurons did all the important work and glia were merely structural. Nedergaard believed otherwise. She had observed that astrocytes in culture could transport fluid, and she suspected they role in clearing waste from the brain. She applied for funding. She was rejected. She applied again, rejected again. The idea that the brain had a dedicated waste clearance system separate from the blood-brain barrier was considered unnecessary. The brain, the prevailing logic went, simply recycled its waste internally. There was no need for a lymphatic-like system because the brain was a closed, privileged organ. Nedergaard persisted. She secured a position at the University of Rochester and built a lab focused on astrocyte biology. In 2012, she and her team published the paper that would redefine how neuroscientists, scientists, understand the brain's relationship with its own waste. The key experiment was elegant in its simplicity. They injected a small fluorescent tracer into the cisterna magna, a fluid-filled space at the base of the skull of a mouse, and watched where it went using a 2-photon microscopy. The tracer should have diffused randomly if the old model was correct. Instead, it flowed along precise pathways, tracking deep into the blood vessels, penetrate deep in the brain tissue, and exiting along veins. The pattern was unmistakable. It was a highway. The paper was published in Science Translational Medicine and was met with a mix of excitement and skepticism. Some researcher questioned whether the same system existed in humans. The mouse brain is smooth, they pointed out. The human brain is deeply folded. Would fluid flow the same way across a convoluted cortex? The answer came a few years later when researchers at the National Institutes of Health used intrathecal contrast agents, an MRI, to observe glymphatic flow in living human subjects. The pattern was the same. The highways exist in humans. The plumbing is real, and this is where the science gets both more promising and more sobering. If the glymphatic system is real and if its primary function is clearing metabolic waste, including amyloid beta, then the path to preventing or slowing neurodegenerative disease become clearer. Optimizing sleep depth and continuity becomes a medical intervention, not a lifestyle preference. There are now at least 5 independent laboratories around the world studying the glymphatic system in the context of Alzheimer's disease, Parkinson's disease, traumatic brain injury, and even normal aging. The consensus that is emerging is that glymphatic impairment may be a common pathway, not the cause of each disease, but the mechanism by which multiple pathologies are accelerated. When the brain cannot clear its waste, every pathological process that produces waste runs faster. Consider traumatic brain injury. After a concussion, the brain produces a surge of damaged proteins and cellular debris that must be cleared. The glymphatic system is the only route for that clearance. But a concussion also disrupts sleep architecture, often for weeks or months. The system that is needed most is precisely the system that is impaired by the injury itself. This creates a feedback loop. Injury generates waste, waste impairs sleep, impaired sleep reduces clearance, accumulated waste causes further damage. It is a cycle that may explain why a single moderate traumatic brain injury increases the risk of developing dementia and decades later by a factor of 2 to 4, depending on the study population. Now the evidence calibration, because the 5 mechanisms described here sit at different levels of certainty. The existence of the glymphatic system in humans is well established. The role of aquaporin 4 in precipitating CSF flow is supported by multiple animal studies and correlative human data. The relationship between slow-wave sleep, norepinephrine withdrawal, and glymphatic clearance has been demonstrated in rodents with direct measurement and in humans with MRI and PET. What remains less certain is the direct causal link between chronic glymphatic impairment and Alzheimer's disease in humans. The correlation is strong. The animal models are convincing, but a definitive human trial, one that shows that enhancing glymphatic clearance over years prevents cognitive decline, has not been conducted. Partly because no reliable method for enhancing glymphatic flow in humans has been validated yet. What we do know is that the variables that impair glymphatic flow are the same variables that increase Alzheimer's and risk: sleep fragmentation, sleep apnea, aging, and hypertension and atherosclerosis. Each of these reduces the efficiency of the hydraulic pump that drives CSF through the brain. Each is modifiable to some degree, and each represents a point of intervention that requires no pharmaceutical approval, no clinical trial, and no prescription. They require only that the brain be given the conditions it evolved to need: uninterrupted, deep sleep in a cool, dark room with a functioning cardiovascular system for a full 7 to 8 hours every night. Think about what that means for the average adult in 2024. The CDC reports that 1 in 3 American adults gets less than 7 hours of sleep per night. Among teenagers, the number is closer to 2 in 3. The reasons are structural school start times, work schedules, screen habits, caffeine consumption, artificial light exposure after sunset. Each of these factors individually reduce the slow-wave sleep. Together, they create a population-wide deficit in glymphatic that has never existed in human evolutionary history. There is a study from 2021 that I want to mention because it keeps the scale of the problem usual, clarity. Researchers at Washington University in St. Louis measured amyloid beta levels in the cerebrospinal fluid of healthy middle-aged adults using lumbar punctures over a 24-hour period. They found that amyloid beta concentration fluctuates in a daily rhythm, rising during wakefulness, falling during sleep. The amplitude of that rhythm was reduced in individuals who slept poorly. The waste was not being cleared. It was accumulating, day after day, year after year, in people who felt fine, who had no cognitive symptoms, who were simply not sleeping deeply enough. The rhythm of the glymphatic system is not something you can feel. You can't sense when your aquaporin 4 channels are open. You can't tell, lying in bed, whether CSF is flowing through your interstitial spaces or stagnating. The system operates entirely below the threshold of conscious awareness, which is precisely why it was missed for so long. Anatomists had seen the perivascular spaces for over a century. They had drawn them in textbooks. They had named them the Virchow-Robin spaces after two 9th century pathologists who first described them. But they assumed these spaces were empty, or structural, or irrelevant. No one thought to ask whether fluid moved through them. No one thought to inject a tracer and watch where it went. Nedergaard did. And what she found was that the important cleaningism in your body runs only when you are unconscious, that it is powered by the same slow waves that your brain generates during deep sleep, and that is silently, systematically impaired by nearly every habit of modern life. The brain washes itself at night, but only if you let it. The closing line, when it comes, should be quiet, not triumphant, not alarming, just a simple restatement of what the data shows. Your brain has a washing system that runs on sleep, on the withdrawal of norepinephrine, on the slow waves, learns that your neurons generate when they are not being asked to process the world. The system evolved over hundreds of millions of years, and it expects conditions that your environment no longer provides. You cannot feel it failing, but the amyloid beta accumulates, the plaques form, the cognitive decline follows decades later. The most effective neuroprotective intervention known to science may be the one thing you are least likely to protect: interrupt deep sleep every night for your entire life. Video na hemfa hetom glymphatic hetom zurnau dukfathin by Maiken Nedergaard by Dihelp Rochester, Vaunum 21-2. Hingu so ka king aquaporin fortune tebau astrocyte moza. Jofep ziknautui chekwa monau, vakwunchoi kakchatain u amyloid beta. Guaching na chi huadom chom zikngu so somchum, he locus coeruleus ngung zyphom norepinephrine. Zukngu bizanduan, tiungu kingnian, valauhua lam suizam kanang tanglopne. Mutkoche kote zytik moilin he zuazoilan zukngu vaben Alzheimer. Tongdip kot lai. But now thuzer psych vabandim. You closed your eyes last night and your brain began to wash itself. You did not feel it. You did not know it was happening. But the fluid moved, the channels opened, depend on how deep you slept, how many times not, how much caffeine how many times you woke, how much caffeine was still in your bloodstream, how warm the room was. The system is not forgiving. It runs on a schedule that your ancestor followed for millions of years and that you've likely broken tonight. The data is clear, the mechanism is known. The rest is a choice about what time you turn off the screen.